Well, welcome everyone to this HESI outreach webinar titled The Update on the Development and Validation of the Pig-a Gene Mutation Assay. I'm going to keep everyone on mute during the webinar just to avoid background noise, but we will have opportunities for questions at the end, and I'll be unmuting everyone at that time. So good evening and good morning, depending on where you're connecting from. Thank you all for joining us, especially from our colleagues in Asia. I would like to thank Ayako Takai, our HESI's advisor in Japan, and Cyril Pettit, HESI's executive director, for making this webinar possible. And also I'd like to thank Cindy Nobles for handling the registrations for the webinar. This webinar series of HESI, this outreach webinar series takes place twice a year, and you can see recordings of past webinars as well as this one in the near future on our website, www.hesiglobal.org, and it's located on our multimedia page. So my name is Jennifer Tenier, and I'm the Scientific Program Manager at HESI, the Health and Environmental Sciences Institute. I'm the manager for HESI's Genetic Toxicology Technical Committee, which the Pig-a Assay is part of, and that's the topic for today's webinar. Before we get started, let me briefly introduce our two speakers, Dr. Robert Heflick of the U.S. Food and Drug Administration, and Dr. Stephen Dertinger from Lytron Laboratories. So first off, Dr. Robert Heflick, he is a research microbiologist and director of the Division of Genetic and Molecular Toxicology at the U.S. FDA's National Center for Toxicological Research, where he has had quite a distinguished career. His present research involves the development of approaches to measure and analyze mutations in laboratory animals. The overall goal of these efforts is the application of sensitive and predictive in vivo mutation assays for regulatory purposes. Dr. Stephen Dertinger is currently Director of Research at Lytron Laboratories. A focus area of his research has been the development of automated methods for detecting and studying genotoxicants. Examples include cross-species, blood-based assays of chromosomal damage and gene mutation, as well as in vitro multiplexed assays for discriminating clastogens, antigens, and non-genotoxicants. After Bob and Steve's presentation, we will have time for questions. If you have a question before then, you can type it into the chat window at any time, and you can send it to me, Jennifer Tenier, or you can send it to all participants. As I said, I'm going to keep everyone muted during the presentation just to avoid background noise, but we'll be unmuting everyone at the end for questions. We'll try to allow for about 10 minutes at the end for questions. And if anyone's unable to see the slides online, you can also follow along with the slides that were emailed to you along with the call invitation. So now I'm going to turn the controls over to Bob and begin that portion of the presentation. So Bob, let me unmute you and let me pass the controls over to you.
Okay, Jennifer.
Okay, Bob.
I feel like I've been released from muted. Well, hello, everybody. As Jennifer said, my name is Bob Heflick, and I'm going to provide a short introduction to this seminar. Most of the speaking is going to be done by Steve Dertinger, however, so Steve, get ready. I'm going to try to go through these introductions very quickly to give Steve as much time as possible. But first, I have to do our lawyer's service and whoop, past one, there we go, and show you our disclaimers and disclosures. In my case, the FDA wants you to know that any day is mine, and they don't necessarily subscribe to it. In fact, they shut down their FDA web server this evening, and I think there may be a message there to me, but we'll see. Everything seems to be fine so far. So next slide. Okay, so what does Pig-a have to do with HESI? and vice versa. What does HESI have to do with Pig-a? So as Jennifer said, HESI has a large committee dealing with genetic toxicology called the Genetic Toxicology Committee, or GTTC, which we tend to use all the time. And its purpose is stated on the slide: to improve the scientific basis of the interpretation of results from genetic toxicology tests for the purposes of more accurate hazard identification and assessment of human risk. And about 5 or 7 years ago, HESI decided that was a useful assay that fit into that category, and we were given status to pursue development of the assay. Okay, next slide, I hope. Okay, now HESI has an interesting structure, at least interesting to me. The GTTC is a partnership between industry, government, and academic scientists. And the idea being that you put these people in a room and they all have different perspectives on a problem, and there's some magic synergy that goes on, and voila, problems are solved and progress is made in scientific risk assessment. And amazing enough, in my experience anyway, with the Pig-a assay, this really seems to work. So on the next slide is sort of an outline of what the GTTC looks like, and all the I can use my pointer, right? Yeah. All the work groups are arrayed in a circle around Pig-a, which appropriately enough is right in the middle. Now, Pig-a started out as an offshoot of this quantitative work group shown here in blue. The purpose of the quantitative work group was to develop methods for modeling dose response data that could be useful for risk assessment purposes. And Pig-a, as it turns out, produces very robust in vivo gene mutation assay data that they thought would be useful for their modeling. And in fact, they have used a lot of Pig-a data. Later on, in 2012, it says here, that there was another work group formed on the adoption of new test methods work group. And in fact, they adopted Pig-a as sort of a test bed for trying out their ideas on the best ways of gaining regulatory acceptance of the new assay. So we sort of have interacted with a number of work groups in the GTTC structure over the years. So hopefully on the next slide shows the leaders of the work group. Myself, Steve Dertinger, Bhaskar Galapudi, who's a consultant, used to work for Dow Chemical, and Anthony Lynch, who works for GlaxoSmithKline in the UK. The purpose, stated broadly, of the work group is to develop the Pig-a gene mutation assay into a regulatory test. Okay. Next slide. And the assay was actually first described in 2008, and it was identified very early by HESI as an assay they wanted to try to develop. And over the years, there's been a number of publications that HESI has published that include Pig-a or on the Pig-a assay. I'll just draw your attention to this last one, which will be mentioned later in Steve's comments, Galapudi et al., as a nice summary of the state of the science in Pig-a and where the data gaps lie and what we need to do to gain regulatory acceptance of the assay. So if you're going to start with Pig-a, I'd start with that paper, which was just published in print a couple of weeks ago in Mutation Research. Next slide. Yes. So about 2 years ago, we decided what we need to do with this assay is develop an OECD test guideline for the assay. and that would really ensure regulatory acceptance. So that became the theme of the Pig-a work group at HESI. So we've held numerous discussions in the last couple of years, and usually related to what we perceive to be data gaps that we need to fill to work up to an OECD test guideline, including increasing the robustness of the assay and applying statistical analysis to the data in the assay. And recently, we've done a lot of discussions about the best ways of analyzing the mutations that are detected in the assay when we look at the mutant phenotype. That's the endpoint in the Pig-a assay, and you probably be able to understand that a little better when you see Steve's talk. In 2013, IWGT, the International Workshop on Genotoxicity Testing, teamed up with HESI to hold a major workshop, and the product of that was that Gallipudi et al. Paper that I just showed that got published 2 years later. Recently, the OECD has accepted our proposal that was prepared largely by the Pig-a work group to put the Pig-a assay on their agenda for developing a test guideline, which the work plan calls for that guideline to be approved in 2021. And here's hoping we can make it. Okay, I'm trying to go to the next one. There we go. So, as this is an outreach to Asia, I thought I'd throw in this slide about Pig-a in Asia, because there's been a lot of activity, especially in Japan, on the Pig-a assay. In fact, my lab at NCTR would never have got started in the Pig-a assay if it wasn't for a Japanese scientist, who I hope was on this webinar by this time, Dashiro Miura, who was on sabbatical from Teijin Pharma in Tokyo, and he, with his skills, got the assay started in our lab, and that was in 2006, 2007. And in fact, first 4 or 5 publications, I'm guessing, he was first author on. So we owe a lot to Japan and to Shiro for getting the Pig-a assay started here. And when Dashiro went back to Japan in 2007, he took his enthusiasm for the assay back with him. And I suspect that's rubbed off on a lot of other people there, because the Japanese Environmental Mutagen Society has been sponsoring a very large multi-laboratory trial on their version of the Pig-a assay. The people at Teijin have developed a variant type of analysis that is very interesting, and they're looking at interlaboratory reproducibility in a large trial. Preliminary, the first report of that has appeared in a recent special issue of Genes in the Environment, and I think Steve later is going to present some data from that study. Okay, as I said, there have been many reports on the Pig-a assay published from Japan, and recently we've got our first paper published from China, and I saw on the list of people who were going to attend this meeting at least one name from that paper, so I hope they're there, and congratulations on your publication, and I hope we can see more publications on Pig-a out of China. Okay, I'm looking for my last slide. There we go. And Steve asked me to bring this up. Steve is going to talk about a version of the Pig-a assay that he's developed with his colleagues at Lytron Laboratories. And it's one that's very popular in the US and in Europe. Okay. But that's not the only way to do the Pig-a assay. In fact, there's a number of variants, and we've done many of them at NCTR. And one of the major variants I mentioned just previously is one developed in Japan, and one of the techniques is termed the pig-ret assay. You may have encountered that term. So all these methods are valid for doing the Pig-a assay, as long as they're done properly. So, and also you should know there are kits for doing the Pig-a assay, but you don't necessarily need the kits if you know what you're doing. I mean, you can buy all the reagents, commercial suppliers, and make your own kit if you want. And you can also not do the assay and pay someone to do it for you, because there's several CROs now that have included the assay as part of their testing services. So I think that's my preliminary remarks, and I'm going to mute myself, or Jennifer's going to do it for me, and turn the microphone over to Steve.
Just a minute. Let me unmute Steve. Got quite a long attendee list here, so I need to find his name.
Okay, wake up, Jennifer.
Oh, here he is. There we go. All right, Steve, can you hear us?
I can hear you. Can you hear me?
Yes, I can.
Okay. Thank you, Bob, and thanks also to Jennifer and others at HESI for giving me this opportunity to talk today about in vivo Pig-a assay. Let me see if I can advance the slide. Yep. Okay. So I'm going to start out by describing the Pig-a assay in general terms, including the assay principle, methods, and assay characteristics. We'll get into some typical experimental designs and a case study involving aristolochic acid. I'll talk about some major conclusions from the IWGT report. That's International Workshop on Genotoxicity Testing. And then we'll also get to an update on progress that's being made to address data gaps to support an OECD test guideline. So here is some essential background information. Pig-a stands for phosphatidylinositol complementation group A gene. So we'll use Pig-a for the rest of this talk. The gene product is required in the first step of glycosylphosphatidylinositol anchor synthesis. We'll say GPI anchors for short. These GPI anchors attach several proteins to the cell surface of hematopoietic cells. So in the case of erythrocytes, these include CD59, CD55, CD24. Now, a key concept here, or principle of the assay, is that of these genes, of the several genes that are required to form GPI anchors, only Pig-a is located on the X chromosome. So that means in the case of males, we have one copy. In the case of females, we have one functional copy. This means one hit, or an activating mutational event, can produce a cell surface phenotype. So when I say hematopoietic cells and I say cell surface phenotype, what that means to a lot of people is that's something that would be very compatible with flow cytometric analysis. And that is indeed the analytical platform of choice for measuring Pig-a mutant cells. Here's a schematic that depicts wild-type cells on the left side, mutant phenotype cells on the right side. And here we've got the red blood cell, for instance, lipid bilayer, and up on top is the external exterior of the cell. Here, I hope you can see my marker. This is the GPI anchor that's linking, for example, this GPI anchored protein CD59 epitope. On the other hand, in this other panel, we have a mutant phenotype cell. Pig-a mutation means GPI anchor was not synthesized, therefore not targeting or linking antigens such as CD59 on the cell surface. This other part of the cartoon is illustrating a fluorescent antibody which can be used against something like CD59, and it represents a way to differentially label wild-type cells, so in this case fluorescent cells, versus the mutant phenotype cells, the non-fluorescent cells. Now, I'm going to describe one early, fairly straightforward approach for scoring mutant cells. I think this will help everyone understand the essential features of the assay. So we're starting with anticoagulated blood. We put it through a reagent like lympholyte. That's a Ficol type reagent. And this can be used to deplete the sample of platelets and leukocytes. Now these red blood cell enriched specimens can be incubated with an antibody against a GPI anchored epitope. In this example, anti-CD59, which is phycoerythrin conjugated. And as we saw in the last slide, this differentiates mutant cells, non-fluorescent, from wild-type cells, the fluorescing cells. Now, after we wash out through centrifugation the unreacted antibody, we bring into contact the cells with a nucleic acid dye. For example, cyto13 has been used effectively. Now, this does a couple things for us. It helps us resolve leukocytes or nucleated cells from the red blood cells of interest, essentially allowing us to gate them out, exclude them from analysis. And it furthermore allows us to differentiate the mature red blood cells with no DNA or RNA content from the immature red blood cells. I use the abbreviation here RET for reticulocyte. The immature red blood cells that are new to peripheral blood circulation and still have RNA content. So they're fluorescent with this cyto13 dye. So here are some bivariate graphs to kind of help you understand what we see on the flow cytometer. On the y-axis is that nucleic acid dye. On the x-axis is the antibody against the GPI-anchored fluorochrome. In this case, it's rat blood, and we use to a great advantage anti-CD59 phycoerythrin. So in these upper quadrants, high cyto13 fluorescence intensity is the immature red blood cells, the reticulocytes. In the lower quadrants are the mature red blood cells, no RNA content. And now here on the x-axis, we see very bright cells, PE fluorescing cells with a uniform and very high signal. These are our wild-type cells. To the left, especially in this vehicle control animal, we see very few cells, red blood cells, with low or no CD59 expression. These are our mutant phenotype cells. These are our presumed Pig-a mutants. Conversely, on the right-hand panel, you see a mutagenized rat exhibiting elevated frequencies of these mutant phenotype cells, CD59 negative, that are showing up in the first decade of log fluorescence. So in fact, if we think about this quadrant some more, we actually get 3 valuable measurements from this simple labeling and staining protocol. percent reticulocytes, an index of erythropoiesis function, also thought of as a bone marrow toxicity endpoint. So these are our reticulocytes, upper quadrants, as a proportion of the total RBCs, all 4 quadrants. We also can enumerate mutant reticulocytes, that's the upper left quadrant, as a proportion of total reticulocytes, the upper 2 quadrants. We can get a measurement mutant erythrocytes, or mutant RBCs, I might say. These are irrespective of their age. These are the mutant erythrocytes in the left 2 quadrants, and we can measure that as a proportion of total red blood cells, all 4 quadrants. I'm going to explain in a subsequent slide what I mean by the mutant reticulocytes are a leading indicator of mutation, and we think of the mutant erythrocyte endpoint as a lagging indicator of mutation. I'll get to that shortly. So time constraints are going to prevent me from describing newer, higher throughput methods in detail, but I do want to make you generally aware of them. Recent advances use antibody-coated paramagnetic microbeads and a magnet prior to flow cytometric analysis. So the so-called immunomagnetic separation methodology allows us to evaluate many times more cells for the mutant phenotype. This will be a little bit of a theme in my talk as I'll introduce this concept, and without the time to really go into detail, I'm just essentially going to direct you to more reading on the subject for the very interested reader. So the so-called Mutaflow commercial kit that's available is described in a Mutation Research issue, 2011. A nice approach developed by Japanese scientists, which Bob alluded to. Pig-a assay is described nicely in an Environmental and Molecular Mutagenesis issue, 2011. Here's some bivariate graphs that I hope give you a little bit of an appreciation for what this immunomagnetic separation in conjunction with flow cytometry technology can do for us. The left bivariate is that same labeling scheme we talked about previously, and this is a mutagenized rat, and we did not separate with that immunomagnetic methodology. We just went onto the flow cytometer. So after some considerable time on the flow cytometer, what we have is a lot of wild type cells and some mutants. Even with a powerful mutagen, this is still quite a rare event, especially the mutant reticulocyte endpoint, this upper left quadrant. On the other hand, with the benefit of immunomagnetic separation, in quite a bit less time, we can actually analyze order of magnitude more cells for the mutant phenotype. So this really ultimately is what's giving us a higher sensitivity assay, higher sensitivity in terms of identifying weak mutagenic effects. So now let's look at some actual data and talk about some important assay characteristics. Here we've treated rats with the mutagen ENU at several dose levels for 3 days. That's important for this slide, so keep in mind 3 days of treatment. Blood samples are collected and analyzed at several time points, including one all the way out to day 90. So now on the left panel here, what we've graphed is mutant phenotype reticulocytes. That's expressed per million. It's a little hard to read that label. Versus time on the x-axis. So that red arrow is orienting you about this early treatment, 3 successive days of treatment. And what's maybe hard to appreciate at first glance is the 0 ENU dose is right at the origin, essentially, a very low, consistent value for mutant phenotype reticulocytes at each and every time point, whereas the mid and high ENU dose levels gave us these very robust dose-dependent increases in mutant phenotype cells. I think one thing that I'd like to bring out or really emphasize on this slide is the fact that in the mutant reticulocyte endpoint, we have a maximal value at about 2 weeks post-exposure, and it's one that's maintained for weeks, indeed maintained for months after that very short-term exposure. For the listeners who are familiar with COMET assay or things like in vivo micronucleus assay, you'll appreciate this assay behaves quite a bit different. Whereas those endpoints are rather fleeting, quickly go back to baseline, this is a protracted effect that's maintained for months, at least for ENU. Now let's go over to the right graph where instead of the young population of cells, reticulocytes, we're looking at the total RBC pool or erythrocyte pool. Again, we have these low frequencies at baseline or vehicle control. We have an intermediate and higher response for these two ENU dose levels. And in this case, we don't have a maximal effect at the 2-week mark. It actually takes several weeks beyond that to fully manifest this mutation induction. This is really, kind of the turnover rate of the reticulocytes, days in circulation, and then it's turned over, whereas this reflects the longer time required to turn over the pool of pre-existing red blood cells with low mutation frequency with the newly created red blood cells that are slowly bringing up the mutation frequency. So this is what we mean by leading indicator of mutation, lagging indicator of mutation for the Pig-a assay. Let's talk a little bit more about the persistence of this effect. This plateau we've been noting, or this plateau suggests that the mutation is neutral. That's terminology that's used in the transgenic rodent literature. It alludes to the fact that the mutant cells in circulation are not highly selected for or against. This neutrality has important implications. For one, mutants should accumulate with repeat dosing. And I will note that this accumulation has been confirmed experimentally. Furthermore, the plateau and persistence suggests that mutation is occurring in hematopoietic stem cells and or early progenitors with considerable self-renewal capacity. That's what explains the fact that we can be out months after dosing has occurred, exposure has occurred, and continue to generate reticulocytes with this mutant phenotype. Here's an example of a repeat dose study design that is being used for many of the published Pig-a experiments you can find in the literature. It's a 28-day study. So in this case, I'm showing rats. That's been published on the most. Usually males are treated with vehicle and up to 3 dose levels of test article for 28 consecutive days. The top dose approximates maximum tolerated dose. Some common important time points that have often been evaluated is an early time point, often used to study micronucleus induction, and then shortly after, within a day after treatment ceases, we get blood for a Pig-a reading. A lot of groups will also study other endpoints, for instance, micronucleus formation in that same blood sample. Optionally, some groups have been studying some of these other time points to essentially form a more complete picture of the manifestation time for developing these mutant phenotype blood cells. At least with immunomagnetic separation technology, it's our expectation for groups to be able to read many millions of red blood cells and easily 3 million reticulocytes for the mutant phenotype. That's per animal, per time point. So let's move on to consider data generated at NCTR when they studied aristolochic acid in rats using this 28-day study design. As many of you know, this naturally occurs as a mixture, aristolochic acid 1 and 2. That's what NCTR studied. It's been implicated in a major disease. In fact, it's an IARC group 1 carcinogen, and to this day, it's a herbal remedy whose use continues in some parts of the world. So, as I said, we'll focus on the NCTR data set and especially their 28-day study. In their publication, they refer to the 28-day experiment as study number 2. Dosing for 28 consecutive days, whereas they studied several time points and several endpoints of genetic toxicity. We're going to focus on micronucleus formation on day 4 and day 29, and Pig-a data on day 29. Using flow cytometry, they were able to score or evaluate many cells. So here's micronucleus data for day 4 on the left side here. In the middle, it's the same 28-day study, day 29 time point. So what they thought they might have seen on day 4 at the 11 mg per kilogram per day dose is a slight micronucleus induction. However, it was not observed on day 29, 1 day after dosing ceased. And in fact, on day 4, in an acute study, they also looked at this 11 dose and in fact went higher and saw no evidence of micronucleus formation. So I think it's fair to say they really do not have a reproducible micronucleus effect here in either of 2 studies. As opposed to the nil micronucleus inducing effect, here we're seeing a very marked dose-related induction of Pig-a mutant cells. On the top panel, we have day 29, Pig-a frequency in the reticulocyte population of cells. And in the bottom panel, we also see day 29 mutant phenotype red blood cells increasing to a high level given this exposure situation. I'd like to note that these robust Pig-a responses were evident in other time points as well, and they also observed this Pig-a induction in their separate acute study. And here's a reference if you want to see more details about that experiment. So what lessons can we take from NCTR's work with aristolochic acid? Well, gene mutation and chromosome damage endpoints are not equivalent. Best hazard ID, and in fact, best understanding of genotoxic mode of action comes from studies that involve multiple complementary endpoints. At this point, I'd like to make it clear that we're not suggesting blood micronuclei and Pig-a results form a complete picture or understanding of in vivo genotoxicity. Rather, they nicely supplement assays that consider other tissues. So beyond work coming out of individual laboratories, it's important to know that coordinated interlaboratory validation efforts have been and continue to be performed. So some of this international work has really been providing encouraging data to support the merits and transferability of the assay. I'll point out a special issue that was in Environmental Molecular Mutagenesis in 2011. Also, more recently, some really nice work out of Japan was put together as a special issue of Genes and Environment, volume 36, 2014 publication. In this lower left area, I'm showing some results from a transferability study that involved 13 laboratories, showing good correlation among these laboratories. And on the right panel, I'm showing some nice work out of Japan, several groups who also show a nice portability of the method, ability of new laboratories to pick up the assay and return expected values and time course information. So promising interlaboratory results encouraged IWGT Steering Committee to include Pig-a in their 2013 program. The resulting report has just been published, as Bob mentioned, Galliputi et al., and it is the current issue of Mutation Research. Major conclusions, some of the more major conclusions are outlined here. Most published data are from rats, and most of our experiences with red blood cells versus other types of hematopoietic cells. Results for 40+ chemicals are highly promising. Repeat dose study designs are preferred in the sense that they benefit from that accumulation of mutant cells with subsequent exposures to mutagen. Given low spontaneous frequencies, at least 1 to 5 million RBCs and reticulocytes should be scored per blood sample, and data for the phenotype-genotype link are encouraging, but more data are needed. A special issue of Mutagenesis is dedicated to filling IWGT and also HESI-GTTC identified data gaps. And I'm really just, I think, given the time is expiring, I don't think I'm going to walk you through all this. I'm just going to highlight the fact that this issue and these papers are really all around filling data gaps in support of OECD. So I'd really encourage you, if you're interested in being aware of the latest work, to find these articles. These three and the three on this next slide. So to wrap up, the take-home messages I'd like to impart to you is that flow cytometry-based methods have been developed for scoring Pig-a mutant erythrocytes. Pig-a complements other genotoxicity assays by covering a broader range of DNA damage. Pig-a is highly suited for integration into repeat dosing toxicology studies, thereby refining and reducing animal usage. And work towards an OECD guideline is underway, and Bob will talk to you a little bit about that. Thank you.
Okay, am I back?
Yes, you're back, Bob.
Okay, good. Very nice, Steve, and thanks for the plug about our work. Always appreciated. So I just have one slide to wrap this up, and it relates to what lies ahead of us in the Pig-a world, and that's as far as HESI is concerned anyway, and that's our plans to develop an OECD test guideline, and that's going to be the major emphasis of the work group for the next few years. And as I alluded previously, last month actually, in April, I think it was the 14th or 15th, OECD, the WNT approved our proposal to start activities towards a OECD test guideline. And the first stage there is, let me get my mouse, is to form an expert working group. Now, I'm sure the people in the HESI Pig-a community are available to us to participate in this, but there's actually quite a bit of work to be done to pull this off and work towards a test guideline. So I encourage anyone who's interested in contributing to this, that perhaps is involved with HESI, to give us a call, and we'd appreciate hearing from you. The first stage in this is to prepare the detailed review paper on the assay, and a detailed review paper is just that. It's a massive document detailing everything in great detail about an assay and how it fits into the rest of the world of genetic toxicology safety testing. And we also have to do a validation type study on the data that exists in the literature, and Steve said 40 plus. It's more closer to 50 compounds now, and we're trying to steer people who are working with the Pig-a assay in particular directions, so they test compounds that might be useful for filling data gaps and doing a validation type study. Okay, while this is going on, HESI will be involved as a partner in doing this with OECD, and already we've held a couple of meetings in anticipation of the approval of our proposal through HESI, and the work group is helping with Pig-a database development and web posting. Now, one of the requirements of an OECD test guideline validation is that all the data be publicly available and the protocol be publicly available. So the way to do that in this day and age is to post it on the web. So we're making arrangements to do just that. And we've got a lot of statistical issues to deal with through the for the validation study, so HESI is helping us out there also. So these are all key validation, part of the validation process. So I encourage you once again, if you want to become involved and you feel like Pig-a is something you're excited about, that you give us a ring and maybe we can work you into the mix. Now, OECD has 34 full member countries, including Korea and Japan. China is kind of in a gray area, and but we wouldn't exclude anyone from China because through HESI, you know, you don't necessarily have to be an OECD member. So let us know, and we will hopefully we can put something together. And by 2021, maybe we can have a test guideline. And I'm trying to advance the slide, and nothing seems to be happening. Could you advance, Jennifer? Okay, here's our, there's nothing left actually. Here's our contact information again, just in case you want to get in touch with us. And the last slide is, thank you. And we've got, in theory, about 8 minutes left. So if you have any questions, please speak up. Perhaps Jennifer can unmute everyone. And we'll try to answer.
We're getting a bit of feedback. Okay. So I've tried to unmute everyone. If you have a question, you can speak up or you can type it into the chat window. If you've muted your individual phone, you may need to unmute that in order to speak. Are there any questions or anyone have any comments? Anyone been working on the Pig-a assay that wants to share any of your experiences? We have a quiet group today. Steve, did you have any other comments to make?
Just that it's early for them. Like it's late for us. But yeah, just thanks again for organizing this, Jennifer. And certainly by emails, we'd be happy to answer questions or, you know, kind of help orient people to a deeper level of where we are and where we'd like to go over the coming year.
Okay, we've got a secret message from someone. You can see.
Okay, this is Ayako, and thank you very much, Bob and Steve, for a very nice presentation. And I didn't know that the early stage of the Pig-a work, so many Japanese, especially Dr. Miura, was involved. And I'm really glad, and also there was much contribution from China. And I hope that for the future process of developing OECD guidelines, we can have many people joining from Asia and then work with the people in the US and Europe to make it happen.
Yeah, this is Bob coming back again. I must say I've been involved with developing a couple of assays, including the transgenic rodent assay and the in vitro micronucleus assay, and all these tend to be international efforts. But I must say that the experience with the Pig-a assay has somewhat been unique in that interaction between different countries has been so strong, and the buy-in from different areas of the world has been quite heartening, at least to me, in my advancing years. So. someone's asking a question. We can't quite make it out.
And did you have a message, Bob, in your chat window? I don't see a message. Or Steve, did anyone ask you a question directly?
Asked me to reply in private, so I guess
okay.
I didn't bring it up in public.
Sure, sure.
There's nothing terrible about it. It's just.
Any other questions or comments? Well, we really appreciate your time today, and thank you for joining the webinar. And again, if you have any questions or anything later, please do feel free to send us an email or reach out to us directly. Thank you, everyone.
Yeah, thank you very much.
Yes, Jennifer. Thanks. Everything went incredibly smoothly.
Thank you. Thank you for presenting, Bob and Steve.
Well, we'll talk anywhere, anytime, for any length.
And we will be posting the link to this webinar on the website, and I'll send an email out once that's done so that you can access it in the future.
Okay, I'll probably be shocked by how I sound, but that's okay.
Excuse me.
Yes?
Can you hear me?
Yes,
I can.
Oh, okay. I have a, thank you for your nice presentation. I have a comment.
All right, go ahead. You have a bird, too.
Can you hear me?
Yes.
The statistical issue is the most important issue for the OECD guideline. So I have a comment. Do you have a lot of negative compound produce a negative result?
Well, Steve, do you want to handle that?
Well,
Do you have enough database for negative compound?
right. I would say what we have right now is an unbalanced data set. What we have is primarily genotoxic carcinogens tested, carcinogenic in the hematopoietic compartment, most of these chemicals, not all of them. We have quite a bit fewer presumed negative genotoxicants studied. Maybe we're up to about 10 negative agents. So that is a concern, and that's something that we understand we have to keep working on. So when Bob Heflick talks about, interested groups approaching us and getting some direction, this is one of the conversations to have is, as a group, what we need to do is study more expected negative agents. I didn't talk about some of our work, but what we've done is emphasize mutagenic, non-mutagenic pairs. So things like ethyl carbamates, in vivo genotoxicants versus methyl carbamates, structurally related non-mutagenic pair. So even if we don't have similar numbers, we like to think we'll have a lot of weight or important negatives tested.
Yeah, we've been sort of surprised by several compounds that are usually considered to be negative in blood, specifically the micronucleus assay, because the metabolites, don't get to the bone marrow, but yet the Pig-a assay is sensitive enough to things like nitrosamines to pick up mutagenicity when we really didn't expect that we'd get it. If you do the assay correctly with the robust magnetic separation techniques, you can actually see very small increases that just aren't apparent with the micronucleus assay. So Yeah, we've got about maybe 10 to a dozen negatives, which is not as many as we would like, but it's not unusual for an undirected retrospective performance assay where you're relying on essentially literature data for your database.
Thank you. Thank you very much.
Any other questions?
Hello, can you hear me? Yeah. Sorry. Yeah. I'm Yan Chang from China. Yeah. Very nice presentation. Can you hear me?
Yes. Go ahead.
Okay. Yeah.
We also got Mozart in the background.
Yeah. And I would like to know during your the OECD guidance drafting process, any plan for such kind of collaboration or co-validation to get more database for the prepare for the guidelines?
Yeah, well, that's one of the things. This is Bob again. That we hope HESI. We can work through HESI to encourage people in certain areas where we need data, okay, to fill data gaps. And the committee that we form with HESI and also with the expert working group of OECD perhaps can identify some of these things where labs that are in the testing and can add, animals onto a study perhaps will be able to do something that would really benefit the of getting a test guideline approved. Now, one thing that I'm aware of that could be a problem for some labs is that they treat animals but don't do the. So what you can do is just take the blood at an appropriate time and ship it off to us, because unfrozen blood samples, properly handled, can be shipped overnight, and we can do the Pig-a assay in a laboratory, a cooperative laboratory, very easy to get data out of that, those animals that you wouldn't normally be able to. So if someone's in this situation, please contact us. We hate to lose the data from those animals.
Yeah, great. Thank you.
Anyone else have a question or comment?
Hello, I see Yan trying to say something.
Yeah, thank you.
Yes, go on.
Thank you very much. I finished my questions, and thank you for your answers.
Oh, okay. I think on the screen that you were talking, and you were beginning rather than ending.
Okay.
I can't see your face, so I don't recognize your voice that way.
Yeah. Right. Thank you.
Well, if there's no other questions.
Hi, this is Takafumi.
Ah.
How are you?
Yes.
Thank you for your presentation. I believe your presentation was very helpful for the audience from Asia, and so to understand the history of Pig-a assay and the perspective of the Pig-a assay.